Good morning, children. Today we are going to see the next topic, advancements in modern biotechnology. Previously, we have been seen the conventional methods. Today, we are going to see about the modern biotechnology. So, it involves many different types of processes. Old biological techniques can be replaced by means of these new technique methods. One is genetic manipulation, protoplasmic fusion techniques. Okay, in this very advanced technology is genetic engineering. Okay, what is meant by genetic engineering? Here it is nothing but recombinant DNA technology, or it is named as gene cloning method. It is named as gene cloning method. Okay, so next one, it is also named as recombinant DNA technology method or gene cloning method. It is named as modification and transferring of DNA from one organism to the another organisms, one organism to the another organisms. So in this we have been learnt, that is exchanging of recombination of genes within the homologous chromosomes. It will be named as a recombination. Okay, the same thing, the recombination process can be followed in this. So here it is, the recombination will be carried by means of artificial modern technology. So we will be naming it as a recombinant or gene manipulation technique. Transfer of DNA from organism to the another, one organism to the another. So that process is named as a recombinant DNA technology. This can be carried out by means of many different types of techniques. One is electroporation, gene gun method, liposome mediated or chemical mediated gene transfer, and mainly it is named as micro injection. So these are all the different techniques to insert the recombinant DNA technology. It can be shortly, it is named as rDNA technology or gene manipulation technique. Manipulation is nothing but multiplying the gene which is we are needed. So that is named as gene manipulation techniques. So in this we are going to learn a new process that is named as steps involved in the recombination of the DNA technology. Okay, so these techniques will be carried out by means of 5 steps. Main 5 steps will be involved. So we will be naming it as a steps or events in the recombinant DNA technology. Recombinant DNA technology, 5 steps are involved. So first step is named as a isolation of gene of interest. Isolation of gene of interest and vector DNA. Okay, isolation of gene of interest. Gene of interest means which gene we have been needed. So they will be named as a gene of interest. And another one is named as vector. Vector is nothing but a gene of interest is carried with the help of some other cloning vehicle. So that cloning vehicle will be named as vector. Okay, and second step, cutting the DNA or plasmid vector. Cutting the DNA, there is gene of interest and plasmid vector, vector which is carrying the gene. So they will be named as plasmid vector using restriction endonuclease. Restriction endonuclease, already you know the function of restriction endonuclease is nothing but it is named as molecular scissors. Molecular scissors means cutting the DNA at particular site. So they will be named as restriction endonuclease. It is an enzyme that is derived from bacteria. Okay, and another one, third step, it is named as splicing. Splicing is nothing but joining the gene of interest as well as vector together. Linking together is with the help of the enzyme that is named as ligase enzyme to produce that as a rDNA. rDNA is nothing but recombinant DNA. So that process is named as splicing process. And fourth one, it is named as insertion of these rDNA into the bacterial cell. Insertion of rDNA into the bacterial cell. And fifth one, it is named as multiplication of rDNA into the bacterial cell using PCR technique. PCR is nothing but polymerase chain reaction techniques, making into multiple copies of rDNA. The process is named as PCR. Okay, so these 5 steps are involved for the recombinant DNA technology. Okay, so shortly you will be saying this 5 process. Okay, so first what it is given means that is nothing means first step I have told that isolation. So in that isolation of DNA fragment which contain the gene of interest will be uh, separated or isolated. Isolated means separated. It is named as insert DNA. It is named as insert DNA. Second one, the recombinant DNA, it will be inserted into a fragment or a carrier molecule that is named as vector. This vector will be easily to self-replicate with the host cell. Host cell, which cell we are inserting, into which cell we are inserting, it can replicate themselves. So they will be named as a vector, or it is otherwise named as cloning vehicle. Okay, and next one, selection of transformed host cell that is carrying the RNA. After joining these two, insert DNA as well as vector DNA, you will be selecting a particular cell which has been accepted or carrying that recombinant DNA with the help of the host, that is vector DNA. So that will be selected. Okay, and next one, entire process will be generated either by large amount of DNA when the protein is inserted into the bacterial cell. When the vector is not involved in the desired gene, if the vector does not receive the DNA, it will not be multiplied. Okay, when it is receiving the DNA only, it will be completely multiplied into the host cell. Okay, it can be inserted by means of few processes, that is named as shotgun method, or already I have told you gene gun method. PCR is nothing but polymerase chain reaction, is a common laboratory technique that is used to make a copies of particular region of the DNA. So the particular gene which we are needed, that gene alone will be multiplied into many in numbers. So that is named as PCR technique. It is also short form, it is PCR. Full form, it is named as polymerase chain reaction. Okay, so here this is a picture of the recombinant DNA technology process. So first step, what I told you, isolation of DNA. Isolation of DNA which is gene interest. Gene interest means which DNA we have to be multiplied into many in number. So they will be named as a gene of interest. And vector plasmid or vector DNA, that is isolated from the bacterial cell. So this will be the first step. Second step, using the restriction endonuclease enzyme, will be cut into many fragments. Fragments is nothing but smaller pieces. It will be cutted into smaller pieces. This will be the second step. Third step, it is nothing but splicing. Splicing is nothing but with the help of DNA ligase, this vector DNA as well as gene of interest will be linked together and made into a rDNA, that is recombinant DNA. It is combining again, so we will be naming it as a recombinant, and this will be the third splicing will be the third step. And next one, fourth step, is nothing but insertion of rDNA into the bacterial cell. When we are inserting into the bacterial cell, bacteria have the capacity to multiply into many numbers into few fractions. Okay, so we are choosing the bacteria as a carrier molecule. So insert this rDNA into the bacterial cell. This will be the fourth step. And fifth step is nothing but multiplying of these rDNA into many copies. Multiplying this rDNA into here many copies. So this will be the fifth step. So first one, insertion of DNA, second one cutting of DNA, third one splicing, that is linking of DNA or joining of DNA with the recombinant DNA with the vector, and next one, fourth one, insertion of rDNA, and fifth one is multiplication of rDNA. So this will be the 5 important steps of the recombinant DNA technology. Okay, thank you.